this is Julia Majeri, and this is the Enzyme Lab. This experiment was conducted by me, Julia Majeri, Sydney Brand, John Malone, and Connor Welker. What is an enzyme? An enzyme is a protein that lowers the activation energy needed for a reaction to occur. The enzyme that we looked at during this experiment is called trypsin, and you can find trypsin in the intestines where it breaks down dietary proteins. And it does this by catalyzing hydrolysis of peptide bonds and amino acids. The substrate that we used is called azocasein, and azocasein is a chemically modified protein used to evaluate enzyme activity. During this experiment, we used TCA to stop the reaction from occurring, and our hypothesis is that as the pH of the protein strays away from a pH of 8, the rate of reaction will decrease. We believe that 8 would be the best pH for enzyme activity because trypsin is found in the intestines where it is a pH of 8. The pHs that we are testing during this experiment is pH 5, 7, 8, 9, and 11. The materials that you're going to need during this experiment are the reaction buffers of 5, 7, 8, 9, 11. You're going to need micropipette, clean micropipette tips, tweezers to pick up the tips, azocasein, trypsin, TCA, 24 test tubes, water bath set at 37 degrees Celsius, spectrophotometer, a centrifuge, and 24 cuvettes. For the procedure, we did this 4 times. For 20 test tubes, you're going to add 450 microliters of the corresponding pH. You're going to add the 500 microliters of azocasein and 50 microliters of trypsin. For the negative control group, you're going to, for 4 test tubes, you're going to add 450 microliters of the pH buffer for 8. You're going to add 500 microliters of azocasein and 50 microliters of deionized water instead of trypsin. And this is to make sure that no reaction occurs during that time. Afterwards, you're going to incubate each test tube for 20 minutes at 37 degrees Celsius in that bath. Afterwards, you're going to add 500 microliters of the TCA to stop the reaction, and then centrifuge the test tubes for 2 minutes at 14,000 RPM for 2 minutes. And this is going to separate the supernatant from the pellet. And then you're going to transfer the supernatant into the cuvette, making sure not to pick up any of the pellet. And afterwards, before you start testing the absorbance, you're going to want to blank the spectrophotometer at 40 nms with deionized water. And then afterwards, you can start reading the absorbance of all the cuvettes with the spectrophotometer. For the results, with our outliers, we saw low activity at pH of 5, higher activity at pH of 7 and 8, the highest at 9, and then it goes back down at 11. If we remove the outliers, we see a pH of 5. It's the same, low activity, and then it steadily goes up until the highest activity at a pH 11. We performed a t-test on those results, and we saw high variance at a pH of 9, lower at pH of 7 and 8, and then the lowest at 11 and 5. And so what to our visual observation, we saw that there was there was a large pellet and light-colored supernatant at a pH of 5. There was a slightly smaller pellet and darker supernatant at a pH of 7. We saw a smaller pellet, and for discussion for visual observation, we saw a large pellet at a pH of 5 with a light supernatant. We saw a slightly smaller pellet at a pH of 7 and a darker supernatant. The supernatant pH of 8 looked very similar to pH of 7, but the pellet was smaller. We saw the darkest supernatant at a pH of 9 and a smaller pellet, and then a small pellet at a pH of 11, but the supernatant was lighter than a pH of 9. And what this means, the darker the supernatant, it means the more reaction that occurred from the enzyme. For the absorbance observations, the results somewhat support our hypothesis. However, neither of the charts with or without the outliers, neither of the highest activity was at 8. We saw here that the highest activity was at 9, and here that the highest activity was at 11. Now for the p-value, the only p-value that was significant was at a pH of 5, and that was at 1.4265 E negative 0.7. For our control, our temperature we kept controlled at a temperature of 37 degrees Celsius, and we thought that this was ideal because the group's testing temperature found the most enzyme activity at a pH of 37. Next time, what we would do different is have more pHs to test. We would keep the same amount of trials and just be more careful overall during the procedure. Thank you. This was the Enzyme Lab presented by Julia Mageri. Goodbye.